In this video I'm going to talk about the concept ATP synthesis. In the previous video I discussed in detail about the electron transport system. So what happens in the electron transport system? As the name indicates, in the electron transport system, the electron is transferred from one carrier to another carrier or from one complex to another complex, during which large quantity of energy is released, and that released energy is used for pumping the proton from matrix regions to intermembrane space. So this pumping of proton from matrix to intermembrane space created proton motive force or electrical potential. So one can tell that the energy which is released during the transport of electron is stored or conserved in the form of a proton gradient. As you can see in this diagram, the proton concentration is high in the intermembrane space and it is less in the case of a matrix. But how can you balance it? To make balance, this proton need to be pumped back from intermembrane space to matrix. But you know that the membrane is impermeable to proton, or this inner mitochondrial membrane is impermeable to proton, and proton can be pumped back only through the channels. And that channel which helps in the pumping off or pumping back of proton from intermembrane space to matrix is known as complex 5. Complex 5 is also called as ATP synthase or ATPase or F0 F1 complex or coupling factor. As the name indicates, ATP synthase or ATPase is the one which helps in the synthesis of ATP. Before discussing the actual synthesis of ATP, we'll discuss about the structure of ATPase or ATP synthase. This is a diagram showing the structure of ATPase. This is matrix, this is inner mitochondrial membrane, and this region is intermembrane space. This ATPase or ATP synthase has mainly two parts, that is F1 complex and F0 complex. This is F1 complex and this is F0 complex. F1 complex or F1 headpiece is hydrophilic in nature, because of which it protrudes into the matrix region. So this is the F1 headpiece which protrude into the matrix region, and this F1 headpiece made up of mainly 5 different types of polypeptide, that is alpha, beta, gamma, delta and epsilon, and these 5 different types of polypeptide are at the ratio of alpha 3, beta 3, 1 gamma, 1 delta and 1 epsilon. So in total there are 9 polypeptide and 5 different types of polypeptide, that is alpha, beta, gamma, delta and epsilon. Out of 5, this alpha 3, that is 3 alpha and 3 beta are arranged in the form of a ring of hexamer with the alternating alpha beta subunit. As you can see in this diagram, this is a hexamer ring with the alternating alpha beta subunit. Alpha beta, alpha beta, alpha beta. Out of this 3 alpha and 3 beta subunit, it is only to the 3 beta subunit ADP will come and bind, or it is only in the 3 beta subunit which has a catalytic site for the ATP synthesis. So it is only in this 3 beta subunit to which ADP will come and bind and it proceeds for the ATP synthesis. So that is about the alpha and the beta subunit. Next is gamma, delta and epsilon. As you can see in this diagram, this gamma, delta and epsilon forms a stalk which connects a F1 and F0 complex, or it makes a part of rotational motor mechanism. Means some part of this subunit will rotate when the proton passes through this proton channel. So this gamma, delta and epsilon makes a part of rotational motor mechanism. Out of these 3 subunits, it is actually the gamma subunit which forms a stalk that connects a F1 complex and the F0 complex, and it is only the gamma subunit which will rotate when the proton passes through the proton pump. So that is about the F1 complex. Now we'll discuss about the F0 complex. F0 complex is an integral membrane protein. As you can see in this diagram, this is a F0 complex which is present in the inner mitochondrial membrane. So that's why it is an integral membrane protein. And this F0 complex mainly made up of 3 subunits, that is A, B and C. And these 3 subunits are at the ratio of 1A, 1B and 9 to 12 C subunits. These 9 to 12 C subunits are arranged in the form of ring. As you can see in this diagram, these are the C subunits which are arranged in the form of ring to form rotor ring or proton channel. So rotor ring or proton channel are nothing but the ring that are formed by 9 to 12 C subunits of the F0 complex. As you can see in this diagram, this is the B subunit which acts as a star that connects to the F1 complex. So it is the B subunit which connects the F0 complex and the F1 complex. And it is the 9 to 12 C subunits which makes up the proton channel. So when the proton passes through this proton channel, the C ring or the rotor ring or the proton channel will rotate. In turn, it activates the B subunit or catalytic site of the F1 complex. So that is about the structure of ATPase or ATP synthase. Now we'll discuss about the actual ATP synthesis. Because of the previous step, that is electron transport system, the proton gradient is created between the intermembrane space and the matrix, or the concentration of the proton is high in the intermembrane space when compared to matrix. So protons are ready to move from intermembrane space to matrix through this proton channel of ATP synthase or ATPase. When the proton passes through the proton channel or rotor ring of the F0 complex, the proton motive force which is already created will drive or will brings about the rotation of this C ring of a F0 complex. So when the proton passes through this channel, the proton motive force will bring about the rotation of this proton channel or C ring of the F0 complex. When the C ring of the F0 complex rotate, it brings about the rotation of the gamma subunit of a F1 complex, because the gamma subunit is attached to the C subunit or C ring of the F0 complex. So when the C ring of the F0 complex rotate, it brings about the rotation of the gamma subunit of a F1 complex. When the gamma subunit rotate, it brings about the conformational change in the 3 beta subunit of the F1 complex. So when the gamma subunit rotate, there is a conformational change or structural change or change in the binding characteristics of beta subunit of the F1 complex. So when there is a change in the binding characteristics of beta subunit, ADP and PI, that is inorganic phosphate, will come and binds to this beta subunit. So when there is a change in the binding characteristics of the beta subunit because of the rotation of the gamma subunit, ADP and inorganic phosphate will come and binds to the beta subunit, which is followed by synthesis of ATP and release of ATP. For proton, when it passes through the proton channel or ATP synthase, it will produce 1 ATP, or when 4 proton pumped back from intermembrane space to matrix via ATP synthase, it produces 1 ATP. In the previous video, that is in the electron transport system, you studied that NADH and FADH2 donate electron to electron transport system. When the electron from NADH enters the electron transport system or passes through the different complex, 10 proton pumped from matrix to intermembrane space. When this 10 proton pumped back from intermembrane space to matrix via complex 5 or via ATP synthase, it can produce 2.5 ATP, because when 4 proton passes through the proton channel or ATP synthase, it will produce 1 ATP. So 10 proton can produce 2.5 ATP. Anyhow, in some of the textbook, this 2.5 is rounded up to 3 ATP. Similarly, when the electron from FADH2 enter electron transport system, 6 protons pumped from matrix to intermembrane space, that is 4 via complex 3 and 2 via complex 2. When this 6 proton pumped back from intermembrane space to matrix via complex 5 or via ATP synthase, it will produce 1.5 ATP, and this 1.5 is rounded up to 2 in some of the textbook. So 1 molecule of NADH can produce 3 ATP or 2.5 ATP, and 1 molecule of FADH2 can produce 1.5 or 2 ATP. During this process or during the synthesis of ATP, the beta subunits will occur in 3 forms. The first form is open form, during which ADP and inorganic phosphate will come and binds to the beta subunits. Upon binding of ADP and PI, the beta subunits will occur in the second conformation or second form, that is tight form, during which ADP and inorganic phosphate will combine to form ATP, or ATP synthesis takes place from ADP and inorganic phosphate. And the third form is loose form, during which the synthesized ATP is released from the beta subunit. So these are the three conformation or three state of the beta subunits during the ATP synthesis. Once the ATP is released from the beta subunits, the gamma subunits of the F1 complex will rotate in a reverse direction and it comes back to the normal position. So that is how ATP synthesis takes place with the help of enzyme, that is ATPase or ATP synthase. I hope this video will be useful. If you like this video, please hit the like button and subscribe to my channel. Thank you.